All right, seems like we're live. Hello, everyone. A very warm welcome to the platform of Biologically Speaking webinar series. I'm Shauradip, and I'm going to be your host today. Thank you for your continued support. If you like our work, please don't forget to subscribe to our YouTube channel to get timely updates about the upcoming webinars. Today, we have Dr. Kathy Friedman with us from the Vanderbilt University, Nashville, Tennessee. She is an associate professor and vice chair of the Department of Biological Sciences. Until very recently, she was also the director of graduate studies in the department and did a phenomenal job in that role as well. Kathy is originally from Wisconsin. After getting her PhD from the genetics department at the University of Washington, she joined the lab of Dr. Tom Check
Mhm.
at the University of Colorado in Boulder for her postdoctoral studies. Kathy started her lab in the Biological Sciences Department at Vanderbilt University, where she now serves as the vice chair. Kathy studies DNA damage and genomic stability, especially focusing on de novo telomere addition in the light of double-stranded break repair. She has been an outstanding mentor for many of us at Vanderbilt and is equally passionate about addressing issues of equity and diversity in science. In her spare time, she loves to water ski and find new running routes around the neighborhood. So, without further ado, Dr. Friedman, the screen is all yours.
Great. Thank you so much for that really nice introduction, and thank you for the invitation. Can you see it now as a full screen?
Yes,
Okay,
we can.
great. Perfect. All right, well, we'll just go ahead and get started. So, what makes a chromosome? I think this is something we don't think about necessarily every day. So, a chromosome, at least here in mitosis, consists of two extremely long DNA molecules. And there are several other really important parts of the chromosome. They need to have origins of replication so that they can be duplicated. They need to have a centromere, which is shown here in the pink, so that the two chromatids can be pulled to opposite poles during mitosis. And then they need to have telomeres, which is maybe the part that you think about a little bit less. And the telomeres are the very ends of the chromosomes, and they're shown here in this kind of light blue color because they're being detected by in situ hybridization using a fluorescent probe that recognizes this very characteristic and repetitive sequence that's found at the ends of chromosomes. So what do telomeres actually consist of? Well, they really consist of both the DNA, which again has this repetitive sequence, and I'll point out that telomeres end in a 3' overhang, so the 3' end extends a little bit further than the 5' end. This 3' terminating end is a very TG-rich strand in virtually all eukaryotes that we know of. And then the telomere is bound by a number of different proteins, and it's really these proteins that play the important roles at telomeres. And some of these proteins bind the single-stranded 3' overhang, and others bind the double-stranded DNA. So in humans, we have about 10 to 15 kilobases of this repetitive DNA at the ends of our chromosomes. In yeast, the single-cell microorganism that I'm going to be talking about today, telomeres are much shorter. They're only a fraction of a kilobase. And yet they have a very similar sequence. So again, they're very GT-rich on this 3' terminating strand. So why are telomeres important? Well, what I think is the most important function of telomeres is that they help distinguish normal chromosome ends from broken ends that are in need of repair. So you can imagine, if you think about the end of a chromosome, that it looks just like a double strand break. And yet the cell has to not treat it as a double strand break, because if telomeres become what we call uncapped, that is, they stop providing this kind of protective function, then the cell detects the normal chromosome end as a double strand break, and it starts fusing chromosomes together, thinking that it's repairing that double strand break. Now, that can happen either because the telomeric sequence itself is lost, or it can happen if there's a defect in those capping proteins that we just talked about. And in fact, that's what the case is here. So now when chromosomes fuse together, we now create dicentric chromosomes, so chromosomes that have 2 centromeres. And when those chromosomes attempt to separate during mitosis, you get these so-called anaphase bridges. The chromosomes are being pulled, and actually the DNA will break. And this sets up a series of events that will, in most cases, lead to the death of these cells. So the other function of telomeres, and this is one that you may have heard of, is also to help provide a solution to the so-called end replication problem. So it was recognized, actually originally proposed by James Watson and then a Russian scientist named Alovnikov, that there would potentially be a problem with replicating the very end of the chromosome. And they didn't quite see it this way because they didn't realize at the time that chromosomes ended in a 3' overhang. So I'm going to tell you how we think this end replication problem happens now. So when DNA replication happens, we know that there's leading strand replication, which on that 5' terminating end can go all the way out to the end of the chromosome. But that on the other strand, we're going to have to replicate through lagging strand replication. And that these RNA primers are removed and the Okazaki fragments are processed to generate a complete strand. But at the very end of the chromosome, when that RNA primer is removed, it leaves this single-stranded overhang. And it was actually that was proposed by Watson and Alovnikov as being the source of this so-called end replication problem. And that's actually probably what you learned in a textbook. But since we know that chromosomes end in a 3' overhang, we understand now that actually that's not much of a problem. It just recreates the 3' overhang again. And the real problem is here on the leading strand, because on the leading strand, there are nucleases that act to recreate that 3' overhang. And it's actually that nucleolytic resection that ends up resulting in some sequence loss at the end of this chromosome. And repeated over many cell divisions, this actually results in loss of telomeric DNA that eventually becomes problematic to the cell. So in steps the enzyme telomerase. And so telomerase is a ribonucleoprotein complex, consists of an RNA component and several protein components. And this RNA can serve as a template. So it base pairs with that 3' overhang. So now you can see why it's important to have a 3' overhang at the end of the chromosome. And the protein component is a reverse transcriptase that then uses this RNA template to add nucleotides to the end of the chromosome. And one thing that's just really astonishing about telomerase is that it has an ability to be processive, so it can actually translocate and add another repeat and go on doing that until telomeric DNA is restored. And once that 3' end is extended, then the normal lagging strand replication machinery can come in and fill in the other strand. So you might have heard that our telomeres actually shorten as we age, and this is a consequence of this end replication problem. And so this is a 4-generation picture taken a very long time ago, actually in my family, because this little baby here is now 24 years old. So her telomeres have shortened considerably since this picture was taken. And so if you look in a population, you find that people who are older tend to have shorter telomeres. Okay? And that's actually because most of the cells in our body, most of our somatic cells, do not express the telomerase enzyme, and so our telomeres shorten over time. And obviously this would be a problem for the continuation of the human race if there wasn't a way to counteract that. And in fact, telomerase acts in germ cells and during early development to rejuvenate or replenish that telomere length each generation. So we start with longer telomeres. There are a lot of studies suggesting that telomere length may correlate with lifespan, that people with longer lifespan on average have longer telomeres, and that increased stress might actually result in telomere shortening. So there's a lot of active research in this area to try to understand how telomere maintenance influences longevity. What is clear is that telomere shortening can be associated with human disease. And I want to point out that these are cases where people have inherited defects in telomerase itself or in other genes that affect telomere maintenance. And so we actually know that there are some diseases such as dyskeratosis congenita, for example, which is caused by mutations in the telomerase enzyme itself. So TERK is the RNA component, TERT is the catalytic protein subunit of telomerase. And these are diseases of stem cell dysfunction. So it appears that even though we don't express telomerase in most of the cells of our body, we do express telomerase in our stem cells, and that's important for allowing sort of tissues that have a high proliferative capacity to continue to regenerate over our lifetime. Okay. So I popped up the little thing a little bit too early here by accident, but you might wonder if we have this enzyme telomerase, and if it's a problem for our telomeres to shorten over time, then why doesn't telomerase simply function in all of our cells? And we think the answer is because too much telomerase activity is associated with cancer. And so the lack of telomerase in many of our cells actually serves as a tumor suppressive mechanism, because when a cell begins to divide out of control, the fact that it's limited by its telomere length is a way that prevents tumors from developing. So I kind of think in many ways we're really a delicate balance between cell proliferation in order to maintain just normal body homeostasis and then the development of cancer. So as I mentioned to you earlier, we actually use yeast to understand very basic mechanisms of cell function involving both telomeres and telomerase and then double-strand break repair. So we use yeast for a number of reasons. It grows quickly. It can be stored really easily. That turned out to be a really important thing this year when we had to shut everything down very quickly. We could just throw everything at the -80 and pick right back up again where we left off. It has a small and fairly simple genome that's been fully sequenced for a long time, so since 1996. It exists in haploid and diploid states, so this makes genetics really easy for us to do. We can look at mutant phenotypes easily in haploids, but we can manipulate cells in a diploid state as well. It's been used as a model organism for many years, so there are a large number of sophisticated research tools that are available. And the genome has been easy to manipulate for a long time. So I will say that CRISPR-Cas9 is helping other more complicated organisms sort of catch up in terms of how easy it is to manipulate the genome. But yeast just remains super simple to make mutations in the genome to see what consequence they have. And many housekeeping genes, whoops, I just accidentally jumped out of that. Here we go. So many housekeeping genes are actually also conserved with humans. And so that means that we can study many of these processes in yeast and know that it's applicable to what happens in human cells. Okay. So as Shordeep mentioned, we are really interested in the intersection between double-strand break repair and telomeres and telomerase. And so I want to introduce that idea to you on this slide. So when a double-strand break occurs, which is what I'm showing you here with this lightning bolt, we know that there are a number of different outcomes that can happen. The most common outcome, obviously, is correct repair. And this can happen through homologous recombination, so copying this sequence off a homolog or a sister chromatid. It can happen through non-homologous end joining, which is essentially just a pasting mechanism that ligates those ends back together. Sometimes during these processes, especially during non-homologous end joining, we can get incorrect repair events. So this can create small mutations at the site of the break or even translocations or larger deletions. Obviously, the worst outcome probably is if repair can't occur at all, because that will result in cell death. And the reason it results in cell death is that as soon as you get a break in a chromosome, you start getting resection or degradation of the DNA on either side of the break. And eventually the chromosome will degrade to the point that essential genes are lost and the cell is no longer able to survive. So the last event that can happen is one that you might not have thought of or might not be familiar with, and that is what we call de novo, or new telomere addition. So this is a case where a new telomere gets added by telomerase to the end of this chromosome. And we're very interested in these events. They're quite intriguing because, in a sense, they're a form of DNA repair, but we like to think of them more as DNA damage tolerance because, in fact, they're quite mutagenic, right? We've just lost the entire end of this chromosome. And yet by adding a telomere here, that telomere now plays that capping or protective role that I told you about. And so it stops any further sequence loss. And so we're intrigued by the possibility that these de novo telomere addition events could also potentially play a protective role in the face of otherwise irreparable DNA damage. There's actually a lot of evidence that these kinds of de novo telomere addition events occur in human cells. And so these are just a few examples of diseases that have been shown to result from de novo telomere addition. And some of them are, quite intriguing. You can see here deletions, 44 subjects. So these are cases where there have been independent telomere addition events occurring in a similar region of the genome, suggesting that there might be certain sites that are much more subject to these types of de novo telomere addition events than other regions of the genome. So back to yeast. In fact, we do see these types of sites, which are what we call hotspots of de novo telomere addition. So these are sequences like this one shown here that have a much higher propensity than other parts of the genome to undergo de novo telomere addition. And I'll show you how we determine that experimentally in just a moment. So if you look at the sequence here of this hotspot, it's perhaps not terribly surprising, right? Because we see that this region is very GT rich. In fact, it's quite similar to this sequence here, which is the sequence of a yeast telomere. So yeast telomeres, as I told you, are GT rich on this 3' terminating strand. They don't have quite the perfect repeat that human cells do, but it's some variation of this 1, 2, or 3 Gs followed by a T. And so our hypothesis is, and we're going to talk about this in quite a bit more detail, that telomerase is able to pair with this region here and then add a new telomere. So we call these sites Sertas, for sites of repair associated telomere addition, and that's what I will largely refer to them as for the rest of the talk. So how do we identify these hotspots or these Sertas within the genome? Well, there are really two main ways that we do this. So the way that one of these sites was initially identified was just looking for spontaneous, what we call GCR, or gross chromosomal rearrangement events. So this work was pioneered by Richard Kolodner's group, and what he did was to generate this chromosome arm in yeast. So it has 2 selectable markers on it, CAN1 and URA3, and these actually are counter-selectable markers in the sense that we can select against them. So if we put cells on media containing the drug cannabinine and the drug 5-FOA, 5-FOA is toxic to cells that have the URA3 gene. Cannabinine is toxic to cells with the CAN1 gene. So what we're doing is forcing the cells to lose both of these genes at the same time. And this can happen through a translocation event or through a de novo telomere addition event, as shown here. Okay. And so, Shurdeep, are you still there?
Yes, I'm here. Yeah, yeah.
You came into my lab, so it made me worried that maybe there was a problem and you were coming to try to tell me. So okay.
No. No, we are fine. Yeah.
Good. Well, someone is working hard then on a Sunday morning, so that's great. Okay, so right, so it could be through a translocation event or through a de novo telomere addition event, as is shown here. And so then we can map the location of these events and we can ask where it's happening. So this kind of assay actually identified one of these Sertas on chromosome 5. But there was a big remaining question, which is whether this hotspot, the fact that we see de novo telomere addition here, is actually due to de novo telomere addition. Like, is this really a hotspot for de novo telomere addition per se, or is this due to increased breakage at this site, okay, and not actually due to increased repair? So in order to answer this question, we developed a different assay system. So in this case, we produce the double strand break ourselves rather than waiting for it to happen spontaneously. So we can induce an endonuclease called the HO endonuclease. And this endonuclease we've engineered the strain so that there's only one recognition site for this endonuclease. So it's going to cut right here, about 3 kilobases away from this sequence that we're studying as a hotspot of de novo telomere addition. We can induce expression of this nuclease by plating the cells on media containing galactose. And when we do that, they begin to express the nuclease, they cleave the chromosome, and then they try to repair that break. So if they repair the break correctly, say by non-homologous end joining, the nuclease is just simply going to cut again. And then they can try to repair again. And so the cells stay in this kind of cycle of cleavage and repair until something happens that actually mutates or eliminates that cut site. Okay. So that can be a mutation right at the HO cut site itself, or it can be a larger event, a de novo telomere addition or a translocation that actually loses the entire end of the chromosome. So we have a URA3 marker out here that allows us to select just for those events that have lost the chromosome end. And then we can use PCR to map the region in which the chromosome was repaired. And you'll see throughout the talk that we map to 3 different regions. We ask whether the event occurred in the Serta itself, which you can see here is a tiny little fraction of this total region. Whether it happened centromere proximal to the Serta, which we call the SEN proximal region. And I'll point out that these are haploid cells. And so this, in this particular case, is about an 8 kb region that lies between the Serta and the first essential gene on this chromosome arm. So the cell can't lose anything past this essential gene or it will die. Or the event might occur telomere proximal to the Serta, somewhere between the HO cut site and the Serta. Okay, so that's how we characterize at least roughly where the event happened. And this is what we see when we do that. So among all the colonies where cells grew in the presence of the nuclease and they had lost URA3, we find that in this case, around 25% or so of those cells have undergone an event within the Serta. And again, I'll point out that the Serta is less than 1% of the total region here. So if you do the math, it turns out that the probability of repair is about 200 times more likely at the Serta than it is at any of the other regions here. And when we look at these events, we find that they're almost always de novo telomere addition. So, and I want to really emphasize here, because this is something different that we do in these assays than other people have done in the past, telomere addition at this Serta can occur after several kilobases of resection from the site of the break. So this experiment really proves that the Serta is not at least solely a site where DNA breakage is more likely to happen, because even if we produce the break several kilobases away, we get telomere addition at this site. And it must be that the DNA has to degrade or resect back to this Serta prior to telomerase acting on that end to create a new telomere. Okay. So this first sequence was identified on chromosome 5, but we've since identified almost about 10 of these Sertas. So I just show you a second one here, which is also GT rich. And again, where we see this preponderance of de novo telomere addition events at this particular site. So obviously we became interested then in what's actually important about this GT rich sequence. And one of the things that was quite intriguing is when we started mapping where the de novo telomere addition events actually occur. So here is this chromosome 5, Serta, and I'm just showing you the sequence here. And the arrows indicate where we mapped telomere addition events. So you can see that most of the telomere addition events happen in this region, which we call the core. And in fact, in some other Sertas that we've studied, it's even more pronounced than this, that all the telomere addition events cluster in quite a small region. And notice that this is oriented so that the endogenous telomere is over here on the right. So it's probably not surprising that if we mutate this core sequence, in this case, we just mutated it to the complementary nucleotides, that we lose de novo telomere addition. And I'll point out that even among these events that happen here, only about half of them actually involve telomere addition. So the effect is even more pronounced than it looks in this graph. So that wasn't too surprising. What was much more surprising is that we found that there's this second GT-rich region that's more internal here, which is hardly ever itself the target of de novo telomere addition, but when mutated, has just as pronounced an effect on de novo telomere addition. So we call this sequence the stim for the stimulatory sequence, and I think of it almost like an enhancer for transcription, in that it is able to, in some way, increase de novo telomere addition within the core sequence. And as we've looked at other Sertas, we're finding that they seem to all have more or less the same kind of structure. Interestingly, the sequence in between here is not important, so we can change this sequence to its complement, and we see essentially no effect on de novo telomere addition. But the presence of the spacer is important. If we delete it so that we put the stim and the core right next to each other, this Serta becomes much hotter, okay? So it undergoes a much higher rate of de novo telomere addition, probably so high that it becomes destabilizing for the genome. And so it may be that this spacer sequence here is important to keep the rates of de novo telomere addition low enough to be compatible with genome stability. So what then is happening at this stim sequence? So obviously, these results suggested that there may be a protein binding to this stim sequence. And the protein that we suspected was a protein called CDC13, because CDC13 binds to normal chromosome ends and helps recruit telomerase. And this sequence, of course, well, you can't see it here, but if you look back here, this sequence here is GT-rich and looks pretty similar to a telomere. So sort of the culmination of a number of experiments, we wanted to prove that the role of the stim sequence was to recruit CDC13. And so we did this experiment shown here. So what we did was to replace the stim sequence with 2 copies of the GAL4 upstream activating sequence. This is just a DNA binding domain for the GAL4 protein, okay? And not surprisingly, that alone does not work to support de novo telomere addition, okay? It's the same as mutating the stim sequence. But if we then express the GAL binding domain, the protein that binds to this sequence, fused to CDC13, we can restore de novo telomere addition. And we tried this with a different protein and it doesn't work. So it does seem to be specific to CDC13. And furthermore, it really relies on tethering to the DNA, because here we have just taken a strain that has just a string of A's here instead of the GAL4 UAS, and now recruiting CDC13 does not increase de novo telomere addition. So that suggested this model here, where after DNA damage and the creation of a double-strand break, we know that 5' to 3' exonucleases come to act upon the DNA. So that generates this long single-stranded tail, and it can be many kilobases long. So we think that when the Serta becomes exposed in single-stranded DNA, that CDC13 is able to bind, shown here in the blue, and that can recruit telomerase then to generate a new telomere at this site. So that model actually brought up some interesting questions about how de novo telomere addition might be coordinated with other repair processes in the cell. So I'm showing you here a model of what happens during double-strand break repair. So we know that when that 3' overhang is first produced, that it's bound by the single-stranded DNA binding protein RPA. And RPA stabilizes that single-stranded DNA. The protein RAD52 then comes and actually facilitates an exchange where RPA is removed and RAD51 is placed here to create the RAD51 nucleofilament that is important for facilitating homologous recombination. So based on this, we were really curious how our model with CDC13 recruitment might intersect with this model of double-strand break repair. And so we asked what would happen if we mutated RAD51. And we were honestly somewhat surprised to find that deleting RAD51 actually decreased de novo telomere addition. We thought we might see an increase by removing competition for binding to the single-stranded DNA. But instead, we saw a decrease in de novo telomere addition. So we then looked at RAD52, and I'll remind you that the model here is that RAD52 is needed to form the RAD51 nucleofilament. Okay? So if RAD51 is required for de novo telomere addition, we reason, well, maybe formation of the nucleofilament is required, and therefore RAD52 should be required as well. And once again, we were surprised because, in fact, deleting RAD52 did not have an effect on de novo telomere addition. And this was really pretty puzzling because RAD51 and RAD52 are thought to work together during homologous repair. So then we did the epistasis experiment. So we deleted RAD51 and RAD52, and we found really this kind of surprised us yet again. So in this case, we found that when we deleted both, we actually got the wild type or the same phenotype as the RAD52 deletion. Okay. And we see the same result if we do it at a different Serta. Okay, so it's very reproducible. So that led us to propose this model. So what this genetics seem to suggest is that what RAD52 is doing is somehow inhibiting de novo telomere addition. But that ability of RAD52 to inhibit is normally repressed by RAD51. Okay, so let me just walk you for a second through why we think this is the model. So in the absence of RAD51, RAD52 inhibits de novo telomere addition, so that's what we see here. But if we delete RAD52, then there's essentially no inhibitory activity, and so we just see normal levels of de novo telomere addition. So in other words, RAD52 seems to be doing something bad, but that's normally repressed by RAD51. So it appeared, let me go back here for a second. So given that we saw this in the complete absence of RAD51, it seemed likely that these effects didn't require nucleofilament formation by RAD51. And so here we use some mutations of RAD51 just to confirm that. So these RAD51 alleles here seem to support normal levels of de novo telomere addition. They behave like the wild type allele. And interestingly, these are alleles of RAD51. This is a co-IP here of RAD51 with RAD52. Here's the interaction in the wild type. And these two mutant alleles retain the ability to interact with RAD52. And they don't affect de novo telomere addition, despite the fact that they don't support nucleofilament formation and they don't support homologous recombination. On the other hand, these two alleles, which I can't remember, yes, here, which disrupt the interaction with RAD51, actually result in the same phenotype as a RAD51 deletion. And so this suggested that this inhibitory effect of RAD51 on RAD52 actually requires the interaction between RAD51 and RAD52. Not nucleofilament formation, but just the ability of these two proteins to interact with each other. Okay, so these results led us to propose this model here. So what we suggested, and this is really just what I showed you before, So what we're suggesting is that in the normal situation, RPA binds, Rad52 helps replace RPA with Rad51 to form the nucleofilament, and that the nucleofilament is permissive, essentially, for CDC13 binding, or perhaps CDC13 can compete effectively with Rad51 for binding and initiation of de novo telomere addition. Okay. However, in the absence of Rad51, we suggest that Rad52 is actually abnormally stabilizing RPA on this single-stranded DNA. And this model is really based on some beautiful single molecule data from Eric Green's lab, which shows, in fact, that when Rad52 interacts with RPA, that it seems to stabilize the association of RPA with the single-stranded DNA. But that, interestingly, despite that, when Rad51 is added, that Rad52 can still mediate this exchange of RPA and Rad51. So those interactions seem to match our genetic data well. So we set out to test this model, and I'm going to sort of introduce three different, introduce our hypothesis here, which I just told you, which is that in the absence of Rad51, Rad52 stabilizes association of RPA with DNA, thereby reducing the interaction of CDC13 with the Serta. Okay, that's our hypothesis. So the first prediction that this hypothesis makes that we could test is that levels of CDC13 association with the Serta should correlate with levels of de novo telomere addition in the absence of Rad51 or Rad52. So what I'm showing here, is CHIP, or chromatin immunoprecipitation, looking at the association of CDC13 with the chromosome following a double-strand break. Okay, so up here is the amount of time that these cells have been in galactose. So we can induce a break and wait 4 or 8 hours, during which time resection of that chromosome end is happening. And so we see that in a wild type cell, we see increased association of CDC13 with the break, which we suggest is association of CDC13 with the Serta. Now, in a Rad51 deletion strain, as we'd predict, we see decreased association of CDC13. So that's consistent with our model. And furthermore, and I don't have the arrows pointing at the Rad52, but if we delete either Rad52 or we delete both Rad51 and Rad52, where we know we regain de novo telomere addition, we also regain association of CDC13 with the break. Okay. So these results are consistent with our hypothesis. Our second prediction is that artificial recruitment of CDC13 to the Serta should suppress the loss of telomere addition in the absence of Rad51. So if the problem is that CDC13 isn't being recruited appropriately in the absence of Rad51, maybe we can override that by artificially recruiting CDC13. And I already told you about the system we have to do that using the 2x UAS. And so indeed what we found is that on the left here is essentially what I already showed you. That is, if we express CDC13 fused to the gal binding domain, we get de novo telomere addition. And now we find in a Rad51 deletion strain that there's no difference. Okay, so deleting Rad51 doesn't seem to have an effect when we're artificially recruiting CDC13 here in a way that doesn't require CDC13's single-stranded DNA binding ability. Okay. So again, consistent with our model. And then finally, our third prediction was that if we could mutate RPA to reduce interaction with Rad52, that would suppress the effect of Rad51 deletion. So again, remember the model is that Rad52 interacts with and stabilizes RPA on the DNA. So if we could prevent that stabilization, we predicted we could overcome this. And so again, let me just kind of put this in a genetic context here. So what we're suggesting is that Rad51 normally inhibits Rad52, but that in the absence of Rad51, Rad52 unusually stabilizes RPA on the DNA. and that stabilization prevents binding by CDC13. So we use this RFA1-44 allele, which is a mutation in one of the subunits of RPA, and we found, if you look over here, that in fact this mutation completely suppresses the defect in de novo telomere addition in the Rad51 deletion strain. Again, consistent with our hypothesis. Okay, so just to conclude this, and then I have a super short little second part. So what I've shown you is that de novo telomere addition is stimulated at TG-rich sequences when the initiating DNA break occurs several kilobases distal to where the telomere is eventually added. Okay. So we think that this is something that could happen when a break occurs at just a random location along the chromosome. Rad51 and Rad52 unexpectedly show opposing effects on de novo telomere addition, and this seems to be because Rad51 counteracts a negative role of Rad52, and that this, in order, Rad51's ability to prevent Rad52 from, boy, that's a lot of negatives, to prevent Rad52 from inhibiting de novo telomere addition depends on the interaction between these two proteins. I also showed you that the recruitment of CDC13 to a Serta is reduced in the absence of Rad51, but that we can overcome that by artificially recruiting CDC13. And so we propose that what Rad52 does is to stabilize the association of RPA with single-stranded DNA in the absence of Rad51, thereby reducing the recruitment of CDC13 to Sertas. Okay. So I mentioned briefly earlier that we know of at least 10 of these Sertas now in the genome. And so just wanted to spend a moment telling you what we're doing along those lines. So Katrina No in my lab has developed what we call the Serta test site on chromosome 7. And so what she can do is take any sequence in the genome that we're interested in studying as a Serta. She can put that sequence on the chromosome and then induce HO cleavage and test it. And the advantage here is that we can test any sequence we're interested in exactly the same chromosomal context, so we don't have to worry about any kind of effects of differing sequence or kind of the neighborhood of the Serta. So when we do this for a number of different sequences, here's what Katrina has found. And the red arrows indicate the two Sertas that we've been using, that I've been telling you about. So you can see that we've actually found some sequences that work even better than these initial Sertas. But we also found a couple sequences that look really TG rich and I thought looked like great potential Sertas that actually don't function at all. So we get quite a range. So when the shutdown happened last March and we couldn't be in the lab, Katrina started talking with her friend Annie Hatmaker, who is in Antonis Rokas' lab in our department and does computational work. And they started thinking, like, wouldn't it be interesting to see if there are any sequence motifs in common between these Sertas? And so Annie and Katrina started with these 3 Sertas. And indeed, they did find that there were some sequence motifs in common. And I'm not showing you the exact sequence here, just in these colors, but they're TG rich, not surprisingly. Okay. And what was really exciting was that when they then looked at the other Sertas that we knew of, they found that every single one of them had at least 2 of these motifs. And maybe even more excitingly, when they looked at the 3 we knew of that don't work, that there was never more than 1 motif matching within these sequences. So it looked as if we had maybe really identified a kind of a sequence signature for those sequences that can function as a Serta. So over the summer, an undergraduate student, Sim Plotkin, wrote a program to identify across the yeast genome everywhere where these motifs lie in close proximity to each other. So what that has yielded is a prediction of about 170 of these sequences across the yeast genome. So they're not terribly common, but there's a good number of them. Some of them are what I'd call simple. So there's just 2 or 3 of these motifs, and others are clustered where there's a very repetitive sequence, and we see many of these motifs in a row. We don't see any obvious bias in terms of the strand these are located on or where they're located in terms of sort of non-essential or essential regions of the genome. But when the motifs do co-occur, there's an extremely strong bias for them to be on the same strand, which we don't fully understand yet. Almost all of them are intragenic, so they're in genes, but a lot of the yeast genome is transcribed, so that's maybe not terribly surprising. What is interesting is that 2/3 of them lie on the template strand. So the GT rich is on the template strand, and that is likely a significant bias that we need to try to understand. And interestingly, some of you might be wondering, we don't see that they're obviously associated with G4 or G quadruplex forming sequences. So it's not clear that these sequences that serve as Sertas are always those sequences that can form G quadruplex. So we've started looking at all of the identified Sertas on chromosome 14, just as kind of a starting point. And I'll say that of the first 3 that we've looked at, they all, in fact, support de novo telomere addition. So I think that this is going to turn out to have been a way that we can finally really identify these kinds of sequences across the whole genome. Okay, and with that, I think my time is up. So I want to thank the people who have done the work. So this work was really initiated by Udo Obodo when she was a graduate student in my lab. The work that I told you about RAD51 and RAD52 was done by Esther Apum, who's now a postdoc at Brandeis with Jim Haber. Katrina No is a current graduate student in the lab doing this work, and I mentioned that she and Annie Hatmaker have collaborated on the computational project, and Annie is in Antonis Rokas' lab. And then this is I had to grab a zoom shot, but this is a picture of Sim Plotkin, the undergraduate student who's been very involved in this project. And I just wanted to point out that we actually, I don't think it's even in PubMed yet, but it should be soon. We just had a mini review article about de novo telomere addition published in Frontiers in Cell and Developmental Biology, and I'm excited about it because you can actually go read about these kind of interesting sequences and some of our hypotheses about how they're being regulated. Okay, so with that, let me get out of my show and get to where I can see people. There we go. I'm happy to answer any questions.
Thank you, Kathy. That was a lovely talk. And I was really impressed with the work that Katrina and Annie put together with this. And it's like a perfect balance of wet lab and dry lab where you like learn something from the wet lab and then you go back, you make a program which actually predicts the Sertas in a better way. So, I mean, that was really good.
Yeah, thank you. I agree. And it's one of those little silver linings. We'd been wanting to do that, but getting forced to be out of the lab for a little while actually put it at the top of the priority list. And then it's turned out to be kind of stunningly successful. I had proposed to do that, and I really wasn't sure we'd be able to find anything. And the fact that we can now potentially predict the distribution of these sites in the genome is really exciting.
Yeah, honestly, I knew about this work obviously from Katrina, like when it was happening, but I didn't know how it turned out. So like that was one question that I was gonna ask you, but like you already answered that one. But we have a couple of more questions. I'll take them. So Dr. Khan is asking, are telomeres common in plants and animals? I think I'll just add one more of my own, which kind of builds on this. So what about the organisms where there is no canonical telomeres? Like, for example, Drosophila that I work with. Yeah.
Yes, absolutely. I know. I'm sorry to have left out your organisms. Yes. So telomeres definitely are common in plants and most animals. So, and in fact, my colleague, Dorothy Shippen, studies, and others too, but studied telomeres in Arabidopsis as a model system. And Arabidopsis has telomerase. And amazingly, when they mutate the telomerase enzyme, telomeres shorten over time, and the plants get smaller and smaller and smaller and smaller until they can't grow anymore. Very similar to what we see in yeast, where we get cells grow more and more poorly as telomeres shorten, or we see in human cells as well. Okay, so Drosophila is an interesting situation, and it is very ironic that actually H.J. Muller was one of the first people to identify telomeres. He actually cloned, or he came up with the term telomere. It means the Greek telos and meros for end part. And he discovered it because he was doing x-rays, using x-rays in Drosophila to generate chromosome rearrangements. And he noticed that the very end of the chromosome was never involved in those rearrangement events. And we know now that that's because the end does, is protected in a way that other double strand breaks are not. However, as we started to understand the molecular nature of telomeres, it became clear that Drosophila don't have traditional telomeres the way that, and it's a subset of dipterans, but as far as I know, it is really restricted to, that set of insects. And the way that telomeres are maintained in Drosophila is that there's actually a transposable element, so a piece of DNA that can move in the genome, that can move from one region and then insert near a telomere. So the chromosome shortens for a while due to the end replication problem. And then instead of using telomerase to replace the DNA, a transposable element moves there and just kind of adds a whole extra chunk of DNA. And then that can go on again for a while. So very, very different. And like I said, very ironic that that's where telomeres were first discovered, and yet anywhere else.
All right. We have Vani. Vani is also part of our platform. So she's asking, can we tweak the property of telomerase DS break repair in silencing cancer's genes?
Yeah, no, not a naive question at all. And I think I can come at that from two different angles. So one is just the role of telomerase more generally in allowing cells to grow in an immortal way. So 90% of human tumors have reactivated the telomerase enzyme. Okay? In order for the cells to continue to grow, they need to maintain their telomeres in some way. And most of the time, that's through expressing the telomerase enzyme. And so there's been a lot of interest in drugs for cancer treatment that would inhibit telomerase. Unfortunately, we don't have a lot of success with those yet, not because we can't inhibit telomerase, but because of this balance I told you about, right? So it turns out that when we inhibit telomerase, they started seeing issues with patients in like hematopoietic cells, for example, so cells that need to be maintained, that need to divide frequently. And so it doesn't look like just sort of outright inhibition of telomerase is going to be helpful. What is looking more promising is targeting telomerase in immunotherapy, because tumor cells are expressing a lot of telomerase, and apparently we don't generally recognize it as a self-antigen. And so you can immunize people against telomerase, and those antibodies will attack the tumor cells. So that is an area which is more actively being addressed. In turn, this question is a little bit more about double-strand break repair. I think we don't know enough yet. It's pretty clear that these kinds of events happen in tumor cells and are part of what gives rise to the genomic instability. How much we can, modify that, I don't know yet.
All right. Thank you. We have another question from Sean Cherry. She is asking, is this Denovo telomere addition double-strand break causing mutation of certain genes and also loss of some genes, because some of those telomere addition will protect this mutated chromosome? And then she added, if it is so, then how it can be overcome?
Yeah, no. So this is a great question because this gets at something we are extremely interested in, and that I kind of alluded to at the beginning of my talk. Okay, so de novo telomere addition is clearly bad because it results in loss of the end of the chromosome, and I showed you some human diseases where this has happened. So it could be that these are simply sites of genomic instability, kind of like fragile sites in chromosomes that we know are sites that break more easily. And it could be that they've been retained because there's some other, this sequence is playing some other role that kind of overcomes this deleterious effect. But the other really intriguing possibility is that maybe they can serve as a mechanism of DNA damage tolerance, which I think is what you're suggesting. Could it be that these sequences in some cases have evolved to allow the chromosome to be saved when there's a break through addition of a de novo telomere? Now, you'd have to suggest that maybe that DNA could be regained again in the future through homologous recombination. And so that's actually one reason we're so interested in understanding how these sites are distributed in the genome, right? Because in yeast, if they're playing this kind of protective role, we might expect a bias toward finding these sequences out toward the end of the chromosome where the sequences are non-essential. We're still working out, I told you there's no obvious strand bias. There might be a slight strand bias, and we're still kind of trying to work on whether that's real or not. So I think the jury's still out on this one, but I think evolutionarily, it's a really fascinating question about whether these sequences are just bad or whether they may actually sometimes be playing a positive role.
On that note, I think we had a question on the same line from Deepak Kumar Poria. He's asking, is there an evolutionary pressure, positive or negative, for this pathway? Is there any evidence this pathway might have influenced chromosomal number in the evolutionary scale?
Oh yeah, that's another really good question. I don't know of specific evidence, but so in other words, do we see cases where you've got related species, for example, where you can see evidence of a de novo telomere addition event? I don't know of any of those cases, but that certainly doesn't mean that they don't exist. We haven't looked for that really carefully. Again, ultimately what I'd love to do is find a situation where we can prevent de novo telomere addition and then say compete two strains with each other, one that can do de novo telomere addition and one that can't, under conditions of maybe mild DNA damage, and ask whether one of them wins out. We haven't figured out yet how to get rid of de novo telomere addition without getting rid of telomere addition altogether. So we have to be able to separate those two things. And so, we're kind of always on the lookout for something that would allow us to do that experiment.
That'll be a very interesting experiment.
Yeah. It's something we can do first, right? Because we can differentially label the two strains and just let them fight it out together and see who wins.
Yeah. All right, I think we'll take two more questions and then wrap it up. So we have one question from Dr. Shaunak Shahu, who is also part of this platform. He's asking, what is the advantage of telomere addition at DSBs, and why do cells employ this mechanism over canonical homologous recombination or NHEJ?
Yeah, so thank you for that question. I don't think they do. Most of the time these cells are really they're using homologous recombination or non-homologous end joining. These events are quite rare. I mean, these sites I'm talking about, it happens at a much higher rate than it in other regions, but the overall rate is still quite low. So I think under normal conditions, the optimal approach by the cell is definitely to do correct repair. What I would argue is that these de novo telomere addition events can arise when normal mechanisms have failed, either through mutation or maybe, it's in G1 phase, there's not a homologous sequence. For whatever reason, non-homologous end joining didn't happen. So non-homologous end joining has to happen quickly before you start getting resection. So if you start getting resection, you've kind of closed off non-homologous end joining as an option. And so at that point, if the cell can't do homologous repair, these de novo telomere addition events might become kind of a last ditch effort to save that chromosome before it just is completely degraded. So I really think of it as a backup mechanism, not, sort of a primary repair mechanism.
I see. All right, last question from Vani again. Can presence of different combinations of Sertas inhibit or increase the chances of telomeres or RADs binding? Basically, can Sertas regulate the telomeres activity?
Right. So, I showed you, and I know we're kind of staring at a random slide here, but I showed you. I can maybe actually go back to that here. That when we look at these different Sertas, we find quite a range in what we call their efficiency. Okay, so the efficiency is just, how much do they stimulate de novo telomere addition? And so we are actually actively looking at what are the characteristics of these different Sertas that influence how well they function. And it's likely to be the extent to which they bind CDC13. So we're also kind of making artificial Sertas with different numbers of CDC13 binding sites so that we can address that real directly. So I think in a sense, the answer is yes. The more CDC13 binding sites we add, the better the sequence will stimulate de novo telomere addition. And I think we'll find that there's a point where it causes an inherently unstable chromosome. And that's something else I'm really interested in asking is, at what point does the rate of de novo telomere addition become so high that even in just a normal population of cells, you'll have a high propensity to lose that chromosome end? Because I would argue that that kind of site would be selected against very strongly evolutionarily.
All right, we'll just end with a last follow-up question. I swear this is the last one from Shona. Are the hotspots like Sertas in yeast also identified in eukaryotic cancer cells that may suggest de novo telomere addition acts as a backup pathway for repairing DSBs?
Yeah, no, great question, obviously. So we don't know the answer to that yet. Like I showed you, it's clear that there are cases of human disease where these kinds of events have happened. Was that because it's a backup mechanism or because, that was kind of a, that it's a site of genomic instability? We don't know. Again, it's clear these kinds of de novo telomere addition events to happen in cancer cells. There's no question they result in, it's one of the ways that you can lose, get chromosome rearrangements in cancer. And there's also, I think, an intriguing possibility that's been suggested that telomere-like sequences located in what we call subtelomeric regions, so just internal to a telomere, may really play an important role in this regard. That if you get a chromosome break near a telomere, that could allow a new telomere to be formed before there's significant resection. So we have not looked directly yet in human tissue culture cells, for example, but it's something we're definitely interested in doing going forward.
All right. Thank you, Kathy. Thank you for coming on our platform. That was a great talk. We had a great interaction. So I think we'll end the talk for today now. We are right at the top of 1 hour. So, all right. Thank you, Kathy. And
thank you. Appreciate it.
Thank you. All right. So everyone else, thank you for joining us today. I hope you guys subscribe to us on YouTube and follow us on Twitter to keep updated on whatever happening with us and whoever we're hosting on our platform. Thank you.